Welcome to Cracking Genes with Zebrafish. Hi everybody, this is our team made up of me, Nurul, June, Adam, Huda, Aida and Kirtana. In this video, we are dividing into the world of genetics through one of science's smallest heroes, the zebrafish. From chromosome counts to gene functions, let's explore why this tiny fish makes a big splash in research. Zebrafish, also known as Danio rerio, is a small freshwater fish that plays a big role in science. It's one of the most popular model organisms, especially in fields like developmental biology, genetics, and drug discovery. One reason it's so useful is because of its transparent embryos, which let scientists watch how organs and tissues form in real time. Plus, zebrafish develop really fast, making them ideal for studying how genes work and testing new medicines. And now we are looking at the number of chromosomes in zebrafish, or Danio rerio. So, zebrafish have 50 chromosomes, that's 25 pairs. Out of those, 24 are autosomes, and one pair is for sex determination. They are diploid, meaning they get half from each parent, just like humans. What's cool is, zebrafish don't have typical X or Y sex chromosomes, but they still develop as male or female based on other genes. But did you know around 70% of human genes have a match in zebrafish? That's why they are super useful in genetic research.
Zebrafish might seem small and simple, but they play a huge role in medical research. First, scientists use them to study serious diseases like cancer, heart disease, and brain disorders such as Parkinson. Despite their size, zebrafish have surprising genetic and biological similarities to humans. This makes them incredibly useful for understanding how these diseases develop and behave. Second, zebrafish are great for testing new drugs. Before moving to human trials, researchers can safely study how a drug works in zebrafish. This helps identify which treatments show promise and which could be harmful, potentially saving both time and life. Lastly, zebrafish help speed up the research process. They grow quickly, reproduce easily, and are transparent at early stages, make it easier for scientists to observe what happens inside their bodies. As a result, new medicine and treatment can be developed faster and get to patients sooner. Alright, so today we are gonna take a quick look at the karyotype of the zebrafish or Danio rerio. It's a small freshwater fish that's super popular in research, especially for genetic and development studies. Sorry if the image isn't very clear, but it shows all 25 pairs of chromosomes. As we know, zebrafish have 50 chromosomes in total, that's 25 pairs. Out of these, 24 are autosomes, and there's one pair related to sex determination. But here's the interesting part. They don't have clear-cut sex chromosomes that humans do, no XX or XY. Instead, their sex is influenced by multiple genes and even environmental factors, so it's more complex and still being studied. Why does this matter? Well, understanding their karyotype helps scientists study everything from how organs develop to what happens in genetic diseases. Zebrafish actually share a lot of genetic similarities with humans. So just to sum up, zebrafish have 25 pairs of chromosomes, known as sex chromosomes, and they are super useful model for learning about genetic and human health.
How many genes do zebrafish have? Zebrafish are tiny striped fish often used in science to study genes, diseases, and development. Scientists have fully mapped their DNA, so we know how many genes they have.
Hello everyone, my name is Kirtana Iswaran and I am from SEN course. Moving on to next, we would like to see about total number of genes in zebrafish and also the numbers came from. So first, the zebrafish has around 26,000 protein coding genes, has around 2,000 non-coding RNA genes, has around 1,500 pseudogenes, and the total amount of annotated genes are around 30,000, which contains in zebrafish. Next, These numbers are come from a very big study were published in Nature in 2013. The author of Hovey et al., who first gave us a detailed zebrafish gene count. Next, they found 26,206 protein coding genes, which is very close to the number where humans have. Not only that, zebrafish share around 70% of their genes with humans, where making them a great for medical research. And also, scientists use them to study cancer, heart disease, and also even brain disorders. Howie K. Clark MD Torojo CF ETL in 2013. The zebrafish references genome sequence and its relationship to the human genome, which Nature is 496, 498, and also 503. That's all from me. Thank you.
Pharmaceutical application. Dopamine receptor, serotonin receptor, GABA receptor are used to study brain function, neurological disease like Parkinson, epilepsy, and test psychiatric drug. Next is cytochrome P450 enzyme are involved in drug metabolism study, help evaluate the safety and toxicity of new medicine. Next is cytokines, interleukins, immunoglobulins are used in immune system study and drug development for autoimmune and inflammatory disease. Next is agriculture application. The first is growth hormone, are studied to enhance growth performance in farm fish. Next, insulin-like growth factor, improved muscle development and growth rate to help optimize aquaculture breeding. Next is digestive enzyme, amylase, lipase, and protease to improve feed formulation and nutrient absorption in aquaculture species. Industrial application. The first is fluorescence protein, used in biosensor and environmental monitoring, like detecting pollutant and toxins. Next, liver enzyme, applied in toxicology screening in industrial chemical and environmental contaminants. Next, recombinant protein production. Zebrafish genes coding for human-like protein are exposed in bacteria or yeast for typical protein production or enzyme study. Alright, so here we are looking at how CYP enzyme activity can be visualized using zebrafish. These embryos have been genetically modified to express a fluorescence reporter. Basically, they glow green when the CYP enzyme is active. In the first row, panel A to D, you can see normal light image of the embryos. The second row, panel E to H, show them under fluorescence to detect the green signal. Panel A and E show the wild-type zebrafish, no genetic modification, so no fluorescence. It's our control. Then, in panel B and F, we got zebrafish injected with red CYP2E1. And boom! You can see that bright green glow in F, which tells us that enzyme is active and the fluorescence reporter is working. Panel C and G show a zebrafish without the EGFP gene, so no green signal as expected. But panel D and H, that's the one with EGFP expression. You can see clear green fluorescence all over, showing that the CYP promoter successfully turned on the fluorescence gene. So yeah, using this method, researchers can track CYP enzyme activity live in real time, and it's super useful for studying drug metabolism and toxicity directly in living organisms.
For the critical analysis of zebrafish is number 1, impact in research. Zebrafish have made significant contribution to developmental biology and genetic due to their transparent embryos and rapid development. In disease modeling and drug scanning, zebrafish allow for large-scale and cost-effective experimentations. For number 2 is limitation and challenger have 2. Number 1 is evolutionary distance and number 2 is ethical concern. For evolutionary distance is the organ and immune system of zebrafish differ of those of human, making them unsuitable for studying center complex disease. For ethical concern is zebrafish are often considered ethically preferable, especially in embryonic stage. For number 3 is effectiveness of modern technology. Advancements such as CRISPR, gene editing, and advanced imaging techniques have enhanced the utility of zebrafish. However, these technologies do not eliminate the organism's inherent biological limitations. How are gene and protein used is use of gene and protein in zebrafish research. Scientists can modify gene in zebrafish to observe what happens when a gene is damaged or functions abnormal. Is a gene that controls cell growth, such as the TP53 gene, is altered. Scientists can study cancer. The protein produced for this gene are also studied to understand how they help the body function. The benefit is they grow out quickly. They are similar to human gene and it is easy to observe can change embryos. What methods are involved is number 1 is genetic editing, CRISPR or Cas9. Technology allows researchers to precisely cut and modify a DNA of zebrafish to create mutations in specific gene. This helps in studying gene function and modeling human disease. For number 2 is transgenic, the insertion of foreign genes into the zebrafish genome to produce fish with nutrient or fluorescence markers, which make it easy to observe cell and proteins directly. Number 3 is gene knockdown or morpholino, using morpholino molecules to block the expression of specific gene during early development, allowing research to study the effect of gene loss of functions. For method number 4 is microscopy imaging. Fluorescence and confocal microscopy techniques are used to observe cellular and tissue structure in detail, especially in the transparent embryos of zebrafish. For the last method is gene expression analysis, RT-PCR, and RNA sequencing. These methods are used to measure gene expression level in zebrafish, helping researchers understand how genes are active or suppressed under normal or disease conditions. For the case study, cancer research using the zebrafish. Research using zebrafish to investigate skin cancer melanoma. A gene that causes cancer cell growth in human was manipulated in zebrafish to create a cancer model that closely mimics the disease. Using this model, scientists were able to directly observe how cancer cells grow and spread, which in the fish body.